fortunate today to have Dr. Gordon Bernard here to talk about what is research from the clinical perspective. And he's going to look back in history and talk about his own experience with clinical research.
Thank you, Sam. It's a pleasure to be here to talk to you about research. I've committed the last 30 years of my career to investigating human diseases, in particular in the intensive care unit. And my role here at Vanderbilt is to further build the infrastructure and the support systems for the conduct of high-quality, compliant clinical research. But I thought it would be a good idea to actually go back and talk about what clinical research is about, how did it begin, where are we in the great scheme of things with regard to clinical research. So there are several types of research. Basic research I will not talk about today. This is research that's done on a benchtop or in animals and other kinds of facilities, but does not involve human work. Bench research can include molecular physiology, cell physiology, tissues, and organs. Then, as I mentioned, there's animal research, which sometimes is used to understand human diseases. In fact, most of the time it's used to help understand human disease, but it is occasionally even at the point where human tissues are being implanted in animals to study them better. So certain cell types might be, or certain DNA might be put in animals so that we can understand better how it works in ways that we couldn't do in a living human being. There's translational research, and I'll talk more about what translation is in just a moment, but it's moving those ideas from the bench and the animal studies into humans. Clinical research is what we do most of. These would be randomized clinical trials that are designed to prove that a device or a drug is particularly good and how best to use those tools. And then that would be T1 translational research leads itself into clinical research. T2 translational research is taking the product of clinical research and moving that out into the community, into hospitals and clinics, and employing those findings to patients who need that type of help. There are a lot of impediments to this translation, moving things along from discovery to actual clinical improvements and improvements in human health. And this slide is designed to show where these impediments lie. There are many that are at this T1 level, going from the bench to clinical. Bench researchers don't necessarily work with clinical investigators, so they don't know each other. They don't always know the bench researcher doesn't always know what it means to put something into human beings, so they don't really design their experiments specifically for a path that goes into humans. They're designing their experiments to answer scientific questions, which may or may not facilitate movement into humans. Then once clinical research is completed and results are known, getting those translated into practice is another huge challenge. I've seen numbers quoted like it takes 17 years to go from findings in clinical research to routine practice. It's almost hard to believe that that's true, but there are clear studies, case studies of various interventions that prove that this is true. The most talked about one is the use of a baby aspirin for acute myocardial infarction. So more than 20 years ago, 25 years ago, large international clinical trials proved that if you show up in an emergency room complaining of chest pain and you're given an aspirin versus not given an aspirin, your mortality is dramatically better if you got that baby aspirin early in the emergency room. So here's a treatment that costs virtually nothing, has virtually no side effects, readily can be done, and as recently as 3 or 4 years ago, there were major institutions in this country where only 20% of patients showing up with chest pain were actually getting an aspirin. It just takes a major effort on the part of the medical establishment to make something happen precisely correctly every time, especially in a busy, crazy emergency room with all kinds of other distractions. So, so these are just issues that we have to face with regard to translation. And what we do research all along this continuum at Vanderbilt, and I'll be focusing mainly on this portion in the middle. So clinical trials come from the French word trier, which means to sort, select, or try. And I'm going to go through a few classic clinical trials as they existed back in the 1500s and more recently. This first case study is from Ambroise Paré, a French surgeon. He was involved in the Battle of the Castle Villain. And normal treatment for battlefield injuries at that time was to pour boiling oil on the injury. One day in a very vigorous battle where there were lots of injuries, he ran out of oil. Just simply ran out of the supplies he needed to do the boiling oil treatment. And instead he used eggs and turpentine because that's what he had. I don't know anything about the rationale for why he would have chosen that, but maybe that was truly all he had. And he put those on, and the next morning he went among the injured troops and he looked at the ones that had been treated with oil and the ones that had been treated with eggs and turpentine, and he was amazed to find that the eggs and turpentine, quote, worked so much better than the oil. Well, I mean, any reasonable person would say, well, it was the oil that was killing him, you know, not that the eggs were so wonderful. And you know, we have lots of history like this. You know, it's suspected that George Washington died by being bled to death. Bleeding was a common medical treatment. That when you were sick, one of the things that would help you would be to bleed. And it'd slice the bottom of your foot and bleed blood out of the bottom of your foot. Surely that must be the bad blood. You know, it all settles to the bottom. And then you let that out, and it's like changing your oil. You know, it's just bad oil out. But if you do that often enough, you become anemic and you die. And there were many in royalty and in politics back at that time that were bled to death. They were literally bled to death. So in next move fast forward here a couple of hundred years. I mean, think about it. 200 years later, the next clinical trial's done. Research goes slowly, but it really went slowly back then. Lind in 1747 was on board the HMS Salisbury, a British ship, and he was determined to study scurvy. And he had a notion that scurvy resulted from a dietary deficiency. So he selected 12 patients on board that had developed scurvy. This is a disease where your skin breaks down and you're bleeding gums and bleeding mucous membranes, and it's lethal. And he selected this group to be what he considered to be a homogeneous group. This is a major issue in clinical research. You want the population to be as similar as possible when you start out, so that if you intervene in one component of it and that population responds in some way, it's easier to see that change against the backdrop of the other patients who look the same at baseline. So he developed a notion of having some concurrent controls. That's a new idea, you know, that you actually control things. Remember, the field surgeon didn't have any controls other than that he had the oil where he was using before, but he wouldn't prospectively randomize people to oil versus eggs. But in this case, Lind did. He said, I'm going to give 2 of these people regular C rations. 2 will take vitriol, 2 will take vinegar, 2 will take salt water, seawater, and 2 will be given fruits, and he describes the fruits. And so he started these treatments and observed them for a period of time. And you know, the story is historically well known that the patients who were given fruit recovered very rapidly from their scurvy and did very well. And from then on, ships that were going to be out to sea for long periods of time were outfitted with adequate amounts of fruit for the troops. Now, only 50 years later, Jenner is looking at smallpox. And he develops the notion of a representative sample. He had this idea from the fact that maids who milked cows, and cows had cowpox. Everybody knew what cowpox was at that time. That these maids would get exposed to these cows with cowpox, and they never got smallpox. And so he had this notion that you could use cowpox to, you could inoculate people with cowpox, and that would prevent them from getting smallpox. So his initial idea was to go to a village and inoculate everybody and see if it could prevent that village from developing smallpox. But then he realized that's too big of an undertaking. He didn't have the resources for that. There was no NIH, nobody to go and apply for funds. So he had to just say, I'm going to just pick a few people who are going to be in this village that hasn't been hit with smallpox yet, but it's coming, and immunize all of those, some of those people. And then they would be a representative sample, compare how many of those got smallpox to the rest of the village, which would be the control, and see if that worked. And indeed, those people were protected. Many others in the village got sick. Many of them died, but nobody in his cohort that had been given smallpox, cowpox died. Now, only a few years later, same century, Haygarth uses the first placebo controls. And so here he's looking at these things that are called Perkins tractors. These are metal rods that are designed very prescriptively. They're a certain length, a certain diameter, they're made out of certain kinds of metal, and that these rods were used to treat a whole variety of things like rheumatism, pleurisy, insanity. And the way that these rods were used is that they would beat people on the back with these rods, and it would make whatever that disease was go away. And you know, I'm going to beat this out of you. And so he had trouble believing that these were doing any good for patients. So he created placebo rods, which were, you know, different shape and size and not the same metal and so forth. And then he had patients either treated with the placebo rods or the Perkins tractors as designed properly. And he found that 4 out of 5 patients treated with placebo had relief, in this case of pain, of their rheumatism. These were rheumatic patients, had relief of their pain with these placebo rods. So either it didn't matter what you made the rods out of, it still worked. If you beat somebody on the back, you know, my back hurts so much now, my knee doesn't hurt anymore. Or that it really is, none of this is working. It's not useful at all. And his conclusion was that none of this is working. This whole notion of beating people on the back with rods is not going to cure disease. So then we go now to Amberson, who is a lung doctor. There really were no lung doctors in the early part of the 1900s. They were tuberculosis doctors. There was no specialty called pulmonology, which is what I am now, but pulmonary medicine was an outgrowth of the TB doctors. And so he was looking at tuberculosis, and he conducted the first truly randomized placebo-controlled trial, which is now the current standard for how we approve drugs and develop drugs. He looked at 24 patients in a sanatorium matched for disease severity, and he tossed a coin and decided that 12 would receive drug versus placebo. Now this is, this last step here is not classic at all. This is not what we would do today. I wouldn't come into this room and say, okay, being exposed to this talk is a medical condition. And I'm going to just you know, divide you into 2 groups and then flip a coin as to which group. What I really want to do is not even flip a coin, but randomize each person individually. Because if you divide people into 2 groups and you flip the coin, it could very well be these groups are quite unbalanced. Just by chance, they'd be unbalanced. So you have a better chance of having nice distribution of patients if you do individual randomization. So this would not be typical today, but it was cutting edge at the time as to how you would do things. The coin toss is also, we don't do coin tosses anymore either. So he was able to show that this one of the early drugs for tuberculosis was actually useful, not anywhere near as good as what we'd like and use today, but at least it was progress, because at the time there was no hope for these people. So this is a quote from one of his papers talking about this, in this case, the single blind concept. Double blind means neither the patient nor the treating team knows what you've been randomized to. Single blind usually means the doctor knows what you're getting, but you don't know what you're getting. So he says then, by a flip of a coin, one group became identified as group 1 and the other became group 2. The members were known only to the nurse and me. The patients were not aware of any of these distinctions. So that was a single blind concept. And again, it was cutting edge at the time in 1931, so less than 100 years ago. In 1938, the Food, Drug, and Cosmetic Act required all drugs to be safe. Prior to that, it was not required. You could sell a drug, and it could be murdering people, and you had no regulatory, there was no regulatory authority to stop you from doing this. And there were certainly lots of snake oil being sold out there. In 1952, there was the first U.S. multi-center clinical trial, and this was again for tuberculosis, streptomycin for the treatment of tuberculosis. And in 1962, the FDA, the Food, Drug, and Cosmetic Act of 1938 was amended, and this is often referred to as the Kefauver Amendment. And now it required not only for those drugs to be safe, but they actually had to work, be effective. And anybody, is Kefauver ring a bell with anybody? Is that familiar? This name appears on the courthouse downtown because he was the senator from Tennessee who offered this amendment to the Food, Drug, and Cosmetic Act. And it was based on some real tragic occurrences in Tennessee, which drove him to want to do this around children receiving toxic drugs. So now, where are we now with regard to developing drugs and new treatments? Typically, there's a phase 1. This is a small number of patients using multiple doses, looking at how the drug is absorbed, where it shows up in the urine, in the blood, and so forth, and then issues around toxicity. And we divide phase 1 into 1A, where only normal volunteers are given the drug. This makes sense for drugs that are going to be used widely for, say, I don't know, for headaches or for diarrhea or something like that. Not for drugs that have high toxicity, like cancer chemotherapy drugs. We wouldn't give chemotherapy to normal volunteers to test it there. So in that case, we would do a 1B, where we're putting the drug into disease subjects. The drugs have toxicity, but the patients are dying, and so it's a reasonable trade-off in that situation. In phase 2, there's a larger number of patients. Usually they're randomized at this point, and now you're looking for proof of principle. You want to say, is there any information coming out of this study that would encourage me to believe that this drug is really going to work? So that's what phase 2 is for. And it refines the kinds of things that you would want to do as you move into the definitive trial of phase 3. So many times there'll be multiple phase 2s. You might do a phase 2 and find out, well, we didn't dose quite long enough, or we didn't dose quite high enough, or we had to exclude certain kinds of patients because they didn't do well in this study for whatever reason. All of those things are part of this proof of principle that leads to the phase 3 definitive trial. And then that trial is usually what is used to go to the FDA and get approval, new drug approval, and get a license to sell that drug. Many times in the process of going to the FDA and getting approval, there's a new drug application requires some work in what's called phase 4. And phase 4 is where you expose many more patients. Now the drug is approved and being sold, and you're tracking what happens in thousands, maybe even tens of thousands of patients. Because if there's a side effect that occurs in 1 in 10,000 people, you're probably not going to pick it up in the first 3 phases. You're going to only pick it up when you get down to phases where there are literally tens of thousands of people exposed. So that's all part of the package of developing these things. Once a clinical trial is completed, there are many, many factors that influence whether or not the treatment will actually be put into place. I mentioned the aspirin story a while ago. That should be something that should be readily translated into practice, and yet it wasn't. Here are some of the factors. Previous clinical experience. So if the drug has been out there and being used for other purposes and people are comfortable with it, they might pick it up faster, for example. Clinical relevance of the outcome measure. If a drug saves lives, that's pretty clear. Okay, we'll do that. But if it's, it might cause you to have fewer headaches over the next 2 years, it's harder to see that endpoint. Consistency with previous results. If a study is suddenly positive, but there have been 10 other studies with that same drug that were negative, I don't know what to do with that. The direction of the results. Typically positive studies generate a lot more interest and movement than do negative studies that say this drug's no better than placebo. In that case, I mean, we're talking now about a drug that was already out there and available and being used for something, and a study then shows that there's no effect where the no effect means something for practice because the drug is available and people are using it. And then finally, the cost of the new treatment and the resources necessary to use it. There are some treatments that are I wouldn't say unquestionably useful, but pretty likely to be useful, but they're so expensive they just simply don't propagate out from the site where the initial studies were done. Maybe they will later when the cost comes down or the ability to expand those resources comes down. And then publications, reader beware. Some authors produce inadequate trial reports. They just simply don't do a good job. Journal editors allow those results to be published sometimes, even though they have flawed. And journals like positive findings. A very large, excellently done study that shows that a certain treatment that everybody's using every day doesn't work, doesn't seem to get published anywhere near as easily as a new drug that's going to cure something that is both have the same benefits for society, one's in a positive way, one's in a negative way. So this is going back again to the 1600s. It's a wood carving where there's a drowning victim here and been pulled from the water, and they're trying to dry this patient off, and This man here, it's hard to see in this picture, but he has a cigarette and he is blowing smoke through this tube that's been inserted into the rectum of this drowning patient. And this was considered to be a saving therapy, that if you put smoke into the rectum of drowning victims, it could save their lives. And needless to say, there are probably many other research studies that are blowing smoke as well. And so this was, later gone out of favor. We don't do that anymore. But it was interesting that it was in vogue, just like bleeding people was. And lastly, I just want to go through 2 or 3 slides of a study done here, that and multi-center study, but led by our group here, that shows how some of this all comes together. So this is ventilator management of patients who have sepsis and ARDS, severe infection and acute respiratory distress syndrome, respiratory failure, double pneumonia. There are a lot of names for this process. So on the chest X-ray, this is infiltrates on both sides of the chest, and this is a CT scan showing infiltrates here. And there are components of the lung that are non-functional. There's some that are normal, very small amount, and then there's some that are marginally functional here. We randomized patients to 6 milliliters per kilogram tidal volume. This is the size of each breath that the ventilator delivers. 6 milliliters per kilogram ideal body weight is what you and I are breathing right now, about 400 milliliters per breath. But for some reason in medical practice, 12 milliliters per kilogram was the standard. I have some thoughts about why that is, but I won't go into it for lack of time here. But that is the standard. That is what everyone was using when they supported anybody on a ventilator. And we surmised that that was too big and might be causing injury by overexpanding the lung. So we randomized patients to those 2 treatments. We found that the mortality rate in those with the high tidal volumes was 40%, those with the low tidal volumes was 30%, so that's a 25% reduction in mortality. This slide actually is, I'll digress for a moment and make a point. You'll often hear in a study result there was a certain percent reduction in mortality. Usually they're talking about this 25%. Where do they get 25%? It isn't 40 going down to 30. That's 10%, right? So where's the 25%? The 25% is that of this total mortality, 25% of it, this is 40%. So of this total mortality, 10% of it, which is 25% of the mortality, is reduced. This causes incredible confusion among people. There are studies out there in cardiology, for example, that reduce mortality in half, from 0.8% to 0.4%. I mean, that's probably good for that 1 in a million person that benefited from that, but it's probably not too good if it costs $50,000 to administer that treatment to every patient, because then you're treating a whole lot of people to get one patient benefit. So the implications of this trial, the name of it was the ARMA trial, was that every year 250,000 people with ARDS show up in our ICUs. With this treatment, lower tidal volume, there would be 1.5 million fewer ventilator days, because this also shortened the time you needed to stay on a ventilator. Mortality was 40%, as I pointed out. Now with low tidal volume, it's closer to 30%. That's 25,000 lives saved each year with this thing. And what we're talking about here is a knob on the ventilator, and it's like your volume on your TV. You just turn it down or turn it up. That's all it is. It's amazing. So in summary, important research goes on here at all stages. It's iterative, cumulative. It starts and goes and starts and stops. Ultimately, it results in tremendous benefits to the health of the public. You don't worry about smallpox anymore, right? But people used to, like whole cities would get wiped out by smallpox. And this whole process starts right here. So thank you for coming.